Because the birth of healthy children is a high priority for public health requires all new babies to be screened for genetic, metabolic, and congenital defects. Newborn screening will not produce cures. It is a harm reduction program. The idea is to identify infants that have serious but treatable conditions that, if left untreated, would result in permanent damage and possible death. It is mandated by our federal government, but run individually by all 50 states. Each state decides what conditions to test for. Newborn screening began in the 1960s with a simple blood test. From a prick in a baby's heel, a drop of blood was placed on a piece of filter paper, which would reveal the presence of a condition called phenylketonuria, PKU. PKU causes mental retardation if untreated. The good news is that a special diet is the prescribed treatment. By 2011, newborn screening in the U.S. had expanded to cover 54 conditions, including sickle cell anemia, cystic fibrosis, deafness, and congenital heart defects. As more effective treatment options for DMD expand, there's an increasing interest in adding a newborn screening test for this disorder. Newborn screening raises several questions. What happens with bad news? Is there sufficient counseling and support to go around? Are sufficient resources available to follow up and treat abnormal results? Who's responsible for follow-up and who pays? What happens when a condition is found for which there is no treatment? Public health is fraught with many ethical questions. Birth defects in children can be triggered by parental behaviors before and during pregnancy. Defects can also be triggered by environmental agents known as teratogens, located in utero or in the surrounding air, earth, water, and the built environment. Live bacteria and viruses are one kind of teratogen. Chemicals, prescription and non-prescription drugs, and radiation are another. It is good public health practice to educate young adults and prospective parents about healthy pre-childbearing behaviors and conditions. For example, bacteria passed in utero from a mother to her fetus through the placenta can cause congenital syphilis. Mother and child can be treated with penicillin, an antibiotic, but if left untreated, the disease can progress to heart, brain, organ damage, and even death. A virus, HIV, can be transmitted from mother to child during pregnancy, delivery, and breastfeeding. The mother could simply avoid contracting either syphilis or HIV, both sexually transmitted diseases, by condom use during intercourse. The inconvenience seems a small price to pay for a healthy child. The Public Health Advisory: engage only in protected sex. Chemicals in tobacco smoke harm babies before and after birth. Mothers who smoke during pregnancy are more likely to have low birth weight preterm babies than mothers who don't smoke. Tiny babies born too early are less developed and at risk for multiple health problems. It is common for babies of smokers to have respiratory problems, and they are 3 times more likely to die of sudden infant death syndrome. The Public Health Advisory: don't smoke. A mother's use of alcohol and drugs can affect a developing fetus. Too much alcohol causes fetal alcohol spectrum disorder, an array of physical and mental defects in the child. Drugs, even seemingly harmless prescription drugs such as a sedative to relieve morning sickness and a medication to treat acne, have resulted in birth defects. The Public Health Advisory: no amount of alcohol or drugs is safe during pregnancy. Be cautious about what you ingest. Vaccination is one of the most cost-effective health investments in public health history. For example, routinely immunizing children against rubella provides lifetime protection, so that a pregnant woman won't be at risk for infection. Infection during pregnancy could result in a child with profound deafness. The Public Health Advisory: be current with vaccinations. In 2003, international cooperation resulted in completion of the Human Genome Project, the mapping and sequencing of the entire human genome. It was a historic milestone. Because each individual has a unique gene sequence, the HGP did not reveal the exact sequence of any or every individual's genome. Using genetic material from a small number of anonymous donors, it sequenced a representative sample. It was anticipated that the genome would uncover mysteries behind diseases and provide clues to treatments and cures. It did provide information on many single-cell defects. It also identified genes tied to Alzheimer's, some cancers, and heart disease. Possible treatments and cures may be discovered in the future, but diseases of later life are complex, and the HGP has made us humble. It's provided a glimpse of what we don't know and the challenges ahead. This is a relatively new and exciting frontier. It has a name: genomics. It's the study of how genes act in the body, interacting with environmental and behavior influences to cause disease. Genomics may help us design personalized therapies to target personal genetic defects. On a larger scale, by analyzing thousands of genomes to compare traits they share and where they differ, scientists may be able to learn why certain people get a disease, that is, how variations in genes affect disease. For example, comparing the genetic makeup of children with and without autism, scientists found that those with autism tend to have more added and deleted sections of DNA in their genomes than those who aren't affected. Where might this discovery lead? While genomics is awe-inspiring and even tantalizing, it must be balanced with the down-to-earth mission of public health, to prevent disease in the entire population now and with practical, cost-effective tools within our reach. We begin with responsibility for ourselves, our behavior toward others, and the stewardship of our environment.